Over the course of the next 2 lectures, we'll be covering folate and B12. These are 2 of the 3 hematological vitamins, meaning that when deficient in these, you'll result in having a form of anemia. Folate has 2 main forms in the diet, and it depends on if it's coming from supplements or whole foods. So here we're looking at folic acid. Folate as a class of vitamin is a carrier of 1 carbon unit, so it carries methyls, carbon with hydrogens connected. And in this picture you see the pteryl monoglutamate form. So this would be found in supplements and fortified foods. And what makes it folic acid is that it only has 1 glutamic acid connected to the teichoic acid. The teichoic acid is made up of pyridine ring and para-aminobenzoic acid, or PABA, and human bodies are not able to connect the pyridine ring to the PABA at this methyl group. However, our bodies can make each of the 3 components, the black, blue, and red components. So we call this the oxidized form, monoglutamate form, and again, it's only found in supplements and fortified foods. The other form of folate is what you'll find naturally occurring in plants and other dietary sources. So here you can see I'm adding multiple glutamic acids, and in food, folate typically exists with up to 9 glutamic acids connected to it. So that can make quite a long branched chain connected to the pyridine components. And you'll notice that on the ring structures that make up the PABA and the pyridine, there are some nitrogens and carbons, and those can carry hydrogens in this case, but those can be switched out for methyl groups. So we call this form the polyglutamate form, and it is natural in foods. Because of the way the ring structure of the pyridine is organized, we can interchange different components and change how many hydrogens or methyl groups are connected to the molecule. And this can change the structure and activity of the folate for different pathways in the body. These changes result from oxidation and reduction. So again, remember that folic acid is already in the oxidized form, and then we can further reduce things down to create dihydrofolate and tetrahydrofolate. And so over on the right, you can see this happening where we have our oxidized form of folic acid. Then we add 2 hydrogens along the upper part of the ring. This becomes dihydrofolate. And then we can add 2 more on the lower part of the ring, which then becomes tetrahydrofolate. Now, typically these are actually appearing as polyglutamates, and in this case we just see the R on the end of the molecule representing that glutamic acid chain, but we're just not drawing it because it would take up a lot more space. So the most common forms of polyglutamate folate would be 5-methyl tetrahydrofolate and 10-formyl tetrahydrofolate. And this is telling us a little bit about the location of the methyl or the formyl groups. And we'll look further at that in upcoming slides. Before we transition though, let's explore where these hydrogens are coming from to create di and tetrahydrofolate. Those are being donated by niacin in the form of NADPH. So remember that NADPH is created in the hexose monophosphate shunt, and it is a reduced form of niacin, and it wants to donate hydrogens and electrons, and so this is one of the many reactions where it's able to do that. All right, so zooming in on the structure of folic acid and dihydrofolic acid, we can see that we've added our hydrogens, and we can switch between di and tetrahydrofolic acid by just having those NADPH donations of hydrogen and electron. So you can imagine that there are many, many forms of folate in food, because you can change what is substituted on those pteridine ring locations. You can also imagine that there could be different chain lengths of glutamate residues. And so right now we know there are over 150 forms that occur naturally in our foods, and they typically have a range of 5 to 9 glutamate residues. Folate in food will have to be altered in order to absorb it. We don't absorb it with all those glutamic acids on it, so we have to hydrolyze it down to the monoglutamate form, or folic acid. If you're consuming your folate as folic acid already with only one glutamic acid from supplements or fortified foods, then no hydrolysis is needed. It's already in the form that is readily absorbable. On the previous slide, I mentioned that 5-methyl tetrahydrofolate is a common form. So we can have this glutamic acid chain, but you can see that there's already a methyl group substituted at the 5th location. So this is where 5-methyl comes in. And this particular location for a methyl group is important in a lot of the biochemistry that occurs in the human body. And therefore, that works out well because it's also a major form that's found in our food. And then all we have to do is cleave off any of the extra glutamic acids that are present. So we actually have enzymes that can add glutamic acids and remove them. So there's a polyglutamate synthetase enzyme, which is ATP dependent, which can actually extend the chain length of folates. And we also have the polyglutamate carboxypeptidase, which will hydrolyze or cut off extra glutamic acids. And so this can be helpful for kind of locking things inside of cells, getting it to move in and out of membranes, so removing things to transfer across a membrane, and then adding things back on to stay in a membrane. For folate sources and bioavailability, if you were just sort of thinking of broad best sources, okra is actually a really good source of folate, as is asparagus. But more typically in American populations, we get most of our folate from fortified flours and cereals. So a cereal, for example, All Bran has 406 micrograms per half of a cup, which is not quite double, but getting close to double what's in okra and asparagus naturally. Raw foods are typically always higher in folate than cooked or processed foods because heating and the addition of acids and bases can affect the folate composition. So that's important to know. So faster cooking methods are usually better for retaining folate. Thinking about fortification, fortification of grains in the U.S. didn't become mandatory until 1998. And the rate at which the mandate required fortification was at 140 micrograms per 100 grams of flour. So if you translate that to a slice of bread, you're getting about 2.5 to 3 micrograms per slice of bread. Going back to the idea of cooking and processing, depending on what method that you're using to prepare food, it could lead to losses that are half to 80%. So again, the faster the cooking method, where it's, maybe a stir fry or microwave, which is pretty quick, those are going to lead to less reduction of folate than other methods like roasting, boiling, things like that. Generally speaking, compared to other B vitamins, this is not as bioavailable. So if you're getting folate from natural sources in a mixed diet, you'll absorb about 50% of what you consume. Now, this is for the polyglutamate form. The folic acid form is very bioavailable. So when I say mixed diet for this, I'm really thinking that you're getting more of that folate from natural food sources. Things that could decrease the bioavailability further beyond the 50% would be that our ability to hydrolyze glutamic acids off of the structure can be impaired by alcohol consumption. There are also natural inhibitors in food sources, so beans, legumes, cabbage, oranges can all have molecules inside of them that can sort of inhibit the release and absorption of folate. And zinc deficiency can also impact folate because there are a number of interconversion steps for folate that use zinc as a cofactor. So if zinc's not there, then it can be hard to get the absorbable form of folate. And we'll look at that reaction in a little bit. And just as a reminder, folic acid does not require any digestion. So folic acid will absorb at about 100% and is much more bioavailable than naturally occurring folate from a mixed diet. The polyglutamate form of folate first has to be hydrolyzed by 2-polypolyglutamate carboxypeptidases. That's a really long set of words, so FGCP is often used to abbreviate those specific digestive enzymes or proteins. So these are within the intestinal mucosa, and they're also released in pancreatic juices and bile into the intestinal lumen. So the FGCP enzyme is the one that's zinc dependent. So if you consume your folate from natural sources that have all those glutamic acid chains on them, then this is the reaction. In order to cleave off glutamic acids, you have to have zinc as a cofactor for this enzyme. So if you're zinc deficient, then breaking down natural folate can be hard. And so this would be a place where if you were zinc deficient and while you're trying to get your zinc status back up, you could take a folic acid supplement and then you would absorb that without any trouble at all because you don't need this enzyme for that. In order to transport the monoglutamate form across the intestinal cell surface, typically that involves ATP and sodium cotransport. So that would be at physiologically relevant or lower doses. And then at high doses, passive diffusion is used. There are a number of folate binding proteins or folate receptors found along the intestinal brush border and within cells of most of our tissues. So folate is really critical throughout the body for cellular processes to occur. Folate, after being absorbed, often travels to the liver, and the entrance of folate into the liver cells is mediated by those folate receptors. So it does need a carrier or shuttle transport protein to allow that transfer across the cell membrane. Liver concentrations of the monoglutamate or folic acid form are then converted by the polyglutamate synthetase into the polyglutamate form. So this is sort of hopefully something you've sort of appreciated from the other B vitamins, that we have the vitamins coming in. We usually have to chop things off of it in order to absorb through a membrane, and then we will then add them back on once they're inside of cells. And so this again is the same sort of transport and activation sequence for folate too. So to move through a membrane, we need the monoglutamate form, but once inside of a cell, to become more functionally active, we have that synthetase enzyme to add on glutamic acids again. So this requires ATP to do that, And before the folate would be released into the blood again, it would have to be chopped off back down to the monoglutamate form. The liver actually will hold on to about 50% of all of the body's folate for a variety of reactions, and so it's stored there in the polyglutamate form because that can't escape through the membranes. Looking at the intestinal brush border, we can see that there are a number of transport proteins depicted here, and there are a couple that I want us to focus on more than others. So first of all, we know that we have our FGCP enzyme, which can break down the extra glutamic acid so that we have our absorbable monoglutamate form. And then absorption will often occur through the proton-coupled folate transporter, or PCFT. This one is more prevalent in the intestinal cells. You'll notice that there's a red X on top of RFC, which is the reduced folate transporter or carrier. This was thought to be a main player of absorption in the intestine, but it's actually more active throughout other organs in the body and not so much the intestine. So PCFT is the main carrier protein to allow folate to move into the enterocytes. And then we have MRP, which allows it to then transport out into the blood. So this process can be impacted by a number of things. The absorption of folate can be impacted by pH. So acid, or the presence of acid, can actually increase absorption. There are a number of genetic polymorphisms that also affect transport proteins and conversion proteins to change between different folate forms. So that could affect absorption, usually in a negative way. Dietary inhibitors, things like in legumes and citrus, can impact the food matrix. If we can't break down the proteins surrounding the folate, then it's not going to be as readily absorbable. And then we've already talked about zinc deficiency being important, particularly for the FGCP, polyglutamate carboxypeptidase enzyme, which breaks down those glutamic acid residues. And within the cell, remember that we want this to get to the di and tetrahydrofolate form. So we want to reduce the molecule down by allowing it to gain some extra electrons and hydrogens. And so niacin, or B3, in the form of NADPH, is responsible for that conversion inside the cells. In the systemic blood, you'll often find tetrahydrofolate monoglutamate, 5-methyl tetrahydrofolate, and 10-formyl tetrahydrofolate, all monoglutamate forms. Tetrahydrofolate has 4 hydrogens. 5-methyl has, instead of at the 5 location, the hydrogen is replaced with a methyl group, and then 10-formyl has a formyl group at the 10 location. And then just as a reminder, so transporting in the blood and across membranes is always monoglutamate forms. It's the polyglutamate form that you'll find within the cells. So for folate tissue distribution, folate will enter tissues through the reduced folate carrier. So this was that protein in the intestine that I said didn't play as much of a role in that tissue, but if we're talking liver, muscle, heart, brain, anywhere else in the body, reduced folate carrier is the major tissue transport protein. So the liver will hold up to about 50% of our body's folate, and the forms in which it's collecting folate in is split into thirds, roughly. So tetrahydrofolate will be 33%, 5-methyl tetrahydrofolate is another third, and then the final third is a 5 or 10-formyl tetrahydrofolate. And all of these would be locked in the liver by adding extra glutamic acids. Glutamate residues can then easily be hydrolyzed back off so that these can be exported from the liver and shared with other tissues as well. In the blood, 2/3 of the folate will be the monoglutamate form, which is already bound to a chaperone protein, so albumin is a big carrier for folate. And then the other third will just be free monoglutamate without any carrier. Red blood cells are fairly well endowed with folate, so this is actually important when we think about status. So we can look at red blood cell folate content, and that tells us how our long-term folate status has been. Because folate is only added to red blood cells at the time that the red blood cell is made. And since red blood cells live for about 120 days, that is a pretty decent lifespan. And so then we know that over the last 120 days, if all of these red blood cells have folate in them, that we've been doing good at maintaining our status over time. For excretion, we can excrete folate in both the urine and feces through bile. And the reason that it will be excreted in bile is because when the liver is going through the reabsorption process through the extrahepatic circulation, some bile can be lost in feces, and folate will come out with that. But typically the urine is thought to be a more major contributor to this. All right, so let's transition to folate functions. Tetrahydrofolate, or THF, is the coenzyme form. So we really want those 4 ring locations to have folates or methyl groups. And if we just think about the general function of folate, we think of it as an acceptor and donor of 1 carbon units, or methyl groups. And so this is involved in, or important for, a lot of synthetic reactions. And when I say synthetic, I mean synthesis reactions. So making new things, adding methyl groups, changing chain length, it could be activating something. So this becomes important in amino acid metabolism, particularly regenerating methionine from homocysteine. And we'll learn next week that B12 is involved in that cycle as well. And then also making purines and pyrimidines for nucleic acids. So this would be related to DNA synthesis when cells are multiplying or repairing DNA. On previous slides, I've mentioned a couple of the different major forms of folate, and so I just wanted to point out where those substitution locations are. So if you remember from organic chemistry, you often have to order all of the components of a structure based on their importance, and so you start at 1 and then move around until you count all of the individual elements. And so positions 5 and 10 are some of the major locations where 1 carbon units can be substituted on this molecule. And so if folate is going to accept and transfer a 1 carbon unit, you will almost always see it at the 5th or the 10th location. So here we're looking at 5-methyl tetrahydrofolate. We can also have methylene tetrahydrofolate, where we create a bridge across the nitrogen at the 5 and 10 location. And then I don't have it depicted, but the next one, the formyl group, is where you get a carbon double bound to an oxygen and also connected to a hydrogen. So we can see what that sort of looks like here. So that would occur at either the 5 location or the 10 location. Those are the 3 major forms that we really use a lot in different reactions. And so let's take a look at some of these reactions. So if we're looking at methionine synthesis, this occurs in 2 steps that also involve B12 or cobalamin. So the cobalamin is bound to the methionine synthase enzyme as a cofactor. So B12 is a coenzyme, and it's going to pick up a methyl group and pass it along. So here we have just cobalamin up here, and then it gains the methyl group as we move down, and now it's methylcobalamin. So that methyl group is donated by folate, particularly in the form of 5-methyl tetrahydrofolate. So I call this sort of a hot potato game of passing methyl groups. So folate has the methyl, it gives it to B12, and then B12 gives it to homocysteine, which changes the amino acid structure to methionine. So that the first step is the transfer of methyl from folate to B12, and then the second step is the transfer of methyl from B12 to homocysteine, and then we produce methionine. There are several things about this particular set of reaction steps that are important. First and foremost is related to something called the methylfolate trap. So the creation of 5-methyl tetrahydrofolate, this is an irreversible step. So once 5,10-methylene THF becomes 5-methyl, the only path forward for it is to give the methyl to B12 when it's bound to methionine synthase. Because there's only one enzyme that can do that. The enzyme that produces 5-methyl tetrahydrofolate is methylene tetrahydrofolate reductase. And sometimes people have a genetic defect in this particular enzyme, making it difficult to make 5-tetrahydrofolate. So that could, in essence, allow homocysteine to build up and make it look like we have a folate deficiency. The other issue here is that we may be fine on folate, but we could have a B12 deficiency, and we're making all the 5-methyl tetrahydrofolate in the world, but without B12 as a cofactor at this intermediate step, we still can't transfer methyls over to homocysteine. And so then the B12 is actually masking this and creating the symptoms of a folate deficiency. When we don't have B12 in this way, we call this the folate trap, because folate will ultimately get trapped as 5-methyl, and because there's only this one reaction it can go through, if B12's not there, then all of the folate will, kind of eventually get stuck as 5-methyl tetrahydrofolate. The problem with this is that folate getting trapped feeds back throughout the folate cycle and can also affect reactions where other forms of folate are important. So, for example, 5,10-methylene tetrahydrofolate is involved in thymidylate and purine synthesis, so making DNA bases. So if all of our stuff is getting stuck as 5-methyl, without additional folate coming in, we can't then use these other folate forms to pass on to those other pathways. So just so that you have an appreciation for the flexibility that folate really provides us, all forms are interconvertible except the 5-methyl tetrahydrofolate form. So when folate from the diet enters our bodies, we will activate it through niacin addition of hydrogens and electrons through di and tetrahydrofolate. From there, the tetrahydrofolate has a variety of pathways that it can cycle in and out of. So this outer cycle, there's an inner bit where we can make 4-amino forms. And you see there's, other cofactors involved here that are kind of highlighted in colored boxes. So we've got riboflavin over here, niacin and peach, and then the purple color, this is B6. And then on the outer edges of these inner cycles, we've got that 5-methyl tetrahydrofolate step that's irreversible. And so just to orient you on the last slide, we were talking a lot about homocysteine being converted to methionine through the help of B12 passing that methyl group. And so here we see that reaction depicted in a different way. And it's important to note that MTHFR, the methylene tetrahydrofolate reductase enzyme that turns 5,10-methylene into 5-methyl THF, uses both riboflavin and niacin as cofactors. And the reason that we don't want homocysteine to build up is because in high amounts it can actually lead to plaque formation in the vasculature and is associated with cardiovascular disease. And so methionine is a safer amino acid to have floating around. So this step is really important for heart health specifically. And again, since this is that 5-methyl trap, because this is irreversible, we can get our methyl groups or our tetrahydrofolate groups stuck as 5-methyl THF if B12's not present. And so they're very intertwined, and that'll be an important concept for you to take away from this in the B12 lecture. And so we call that the folate trap, and 5-methyl THF is what builds up if B12 is not present. All right, so let's just take a step back and think about how these different reactions and interconversions relate back to our health and disease relevance. So tetrahydrofolate, its job is to accept and donate methyl groups. This is really important for amino acid metabolism. We've already seen an example where we convert homocysteine into methionine. And this is also true for other amino acids, so switching between histidine, serine, and glycine. So definitely folate plays a role in amino acid metabolism. The other main place that it plays a role is in nucleotide synthesis, so making purines and pyrimidines for DNA bases. So this means that folate is therefore essential for cell division, and this becomes important with red blood cell formation and minimizes our risk of anemias, but also it has an important role in cancer risk. So folate is needed for cells to reproduce and multiply when necessary, but cancer cells also like folate, and so then this becomes a little bit of an interesting conundrum where we have to balance folate metabolism during cancer, and many of our cancer treatments actually try to exploit the folate interconversion steps, which we'll look at soon. But first, let's look at amino acid metabolism. So when we're breaking down histidine, this is one of the amino acid conversions that do use folate, and we can actually use histidine breakdown or catabolism as an indicator of folate deficiency. So we give a person oral histidine, and then we measure the activity of the formaminotransferase enzyme. So we can see that over here. And you'll notice that histidine first becomes urocanic acid, then formaminoglutamate or FIGLU, and then finally converts to glutamate. So if folate is not here as a cofactor for this enzyme, then we get a buildup of formaminoglutamate or FIGLU, and then that will be in the blood, it'll spill out in the urine, and then it tells us, hey, this is building up, so you're probably folate deficient, and now the conversion and breakdown of histidine is disrupted. Folate is also involved in the interconversion between serine and glycine and the further breakdown of glycine into ammonia and carbon dioxide. And so in both cases, we use tetrahydrofolate interconverting between the tetrahydrofolate and 5,10-methylene. So this is where carbon is being transferred. So in serine, it's taking a methyl group from serine and it becomes glycine. And then for glycine to be further broken down, a tetrahydrofolate again will take a methyl group off of it. And so we're basically just chopping up the chain length to make shorter and shorter molecules so that these can then be excreted in urine. For purine synthesis, so making DNA bases, we need tetrahydrofolate as a cofactor at a number of steps. So I've kind of got them highlighted here in blue squares. Oops. And you can see there are a lot of steps involved in creating these ring structures. So we start with a ribose 5-phosphate, which would come from the hexose monophosphate shunt, because remember we make pentose sugars there. And then we cycle through a lot of steps where amino acids are donating components like glutamine, glycine, and those repeat themselves around. We have N-formal tetrahydrofolate up here that's used, and then again we have N10-formal over here as well. And so this is making a precursor inosine monophosphate that can be incorporated into DNA bases. So DNA bases and their involvement of folate and donating methyl groups becomes important in folate and cancer risk. So folate deficiency, that's chronic, can lead to undermethylation of DNA and misincorporation of DNA bases. So uracil is an RNA base that has to be converted to thymine to actually enter DNA. And so if we don't have enough folate, we have misincorporation. So now we have mismatched pairs within the DNA. So that leads to DNA mutations and therefore potentially carcinogenesis. The other thing that we haven't talked about, which we'll really explore with B12, is that without folate to donate methyl groups to B12, we aren't able to methylate our DNA. So when we talked about biotin, we talked about how our DNA can be tightly wound up around histone beads, and that helps with expression of DNA sequences. DNA also can be silenced by adding methyl push pins to the outer edge. So basically telling the cell if it can transcribe a portion of the DNA or not, and methylation of the outer edge of DNA will say, no, we're not going to sequence this part of the DNA. And so this affects gene regulation. And if gene regulation gets disrupted, that can also lead to carcinogenesis. So adequate intake is really important for preventing cancers, but it's a double-edged sword because in cancer patients, because cell division is so important, and every new cell needs DNA, excess folate can actually accelerate the multiplication of cells because it's providing extra folate to keep making all of those new DNA bases. And so folate gets a little bit tricky, and so you don't want too little, you don't want too much. And modern medicine is sort of capitalized off of the involvement of folate in cancer development, and the cycle that goes from methylene tetrahydrofolate to dihydrofolate back to tetrahydrofolate. So this recycling of the different forms of DNA that are then tied to producing DNA bases up here, so taking uracil and turning it into thiamine, these become important. And one of the main leukemia drugs is methotrexate, and it inhibits the dihydrofolate reductase enzyme. So here we're trying to go from dihydrofolate to tetrahydrofolate. Methotrexate stops this step here. And this means that we aren't able to continue through the cycle to make tetrahydrofolate and then N5N10 methylene, which ultimately fuel the production of purines. So what we really ideally want is methylation of uracil, but if the methylene tetrahydrofolate is converted and turned into dihydrofolate, in order to get back there to continue to methylate uracil, we need this dihydrofolate reductase enzyme to work properly. And in cancer, we can actually stop this cycle from happening, and this will basically say, okay, well, cancer cells, you can't incorporate the right bases now into your multiplying cells, and that becomes a problem for the cancer cells. And the hope is that our cells are more resistant and resilient to having this cycle disrupted than the cancer cells. And indeed, this treatment works pretty well for a number of leukemias and other cancers. So particularly childhood leukemia is treated with methotrexate. All right, so let's talk a little bit about recommended intakes for folate. So oftentimes we have to think of dietary folate equivalents because folic acid and food-based polyglutamate form of folate are a little bit different. So folic acid is very bioavailable. We absorb basically 100% of what we consume. So a folate equivalent would be 1 microgram of food folate, 0.6 micrograms of supplemental folic acid, and that includes fortified foods. And if you consume the supplement or the fortified food on an empty stomach, then it's 0.5 micrograms. So an empty stomach leads to even greater bioavailability. So we want people to get 400 folate equivalents per day. So they need fewer from supplemental or fortified foods relative to whole food products. So if we were to translate that, 400 dietary folate equivalents would be 240 micrograms of folic acid from grains or other fortified products. So that's a multiplier of 1.7. And if you just took a folic acid supplement on an empty stomach, then you only need 200 micrograms. And so that's a 2-time conversion rate. To look at folate status, we can look at our plasma levels. Anything above 6.8 nanomoles per liter reflects recent good intake. If we want to know how we've been doing over the last 2 to 3 months, then we look at red blood cell folate composition, and we want to see that above 360 nanomoles per liter. And then, again, those are more static, just telling us about recent and more longer term consumption. We can also look at the functional activity of folate. Deoxyuridine suppression tests are used. So this is looking at that thymidylate synthetase activity. So we culture red blood cells and look at the presence and absence of folate, B12, or both, and seeing how well that enzyme operates within the red blood cells. The other thing we can look at that tells us about function is whether or not we have elevated homocysteine levels. If homocysteine is elevated, that indicates that we may have a deficiency of folate or B12. And so we have to assess for either of those because they can sort of mask one another. If someone is folate deficient, they develop megaloblastic macrocytic anemia. So this is where the cells are really large and often still have nuclei inside of the red blood cells. So anemia is characterized with fatigue, weakness, headaches, heart palpitations. The megaloblastic anemia occurs because we have a reduced synthesis of DNA with an excess production of hemoglobin. And this usually occurs about 4 to 5 months of deficient intakes of folate. Because we're not synthesizing DNA as well in this situation, that low folate could actually increase the risk for cancer due to DNA strand breaks in dividing cells that are not able to repair those breaks effectively because we don't have new bases to add. Now, pregnant women have an increased demand for folate because there's so much extra DNA synthesis going on to fuel fetal development. And folate deficiency has long been associated with poor pregnancy outcomes like low birth weight, fetal growth retardation, congenital heart defects, neural tube defects, which are probably the most commonly known impacts. And that occurs when the neuronal tube is trying to multiply and close itself. And if neuronal cells can't develop and migrate properly, then neural tube defects occur. There are other specific populations that are at risk of folate deficiency. These would be alcoholics, those with irritable bowel diseases, and those with MTHFR enzyme polymorphism. So that, again, is the enzyme that converts and creates 5-methyl tetrahydrofolate. If that enzyme's not working properly, then we aren't able to pass methyls over to B12 and then homocysteine. Let's take a quick look at the process of red blood cell formation. So as red blood cells mature into erythrocytes down at the bottom of the slide, you'll notice that there are precursor cells with nuclei that come together. They merge into one cell, and then eventually the nucleus is degraded. So red blood cells do not have DNA in them once they're mature. If someone develops anemias, particularly folate or B12, we end up with these sort of intermediate erythroblast cells where they still have nuclei, they still kind of have rough edges. They're not perfectly oval shaped like red blood cells. So this is where the megaloblastic anemia component comes in. So if you looked at a blood smear and saw this, you would immediately be able to say, yeah, that's probably a folate or B12 problem. Now, B6, on the other hand, also causes anemia, but it causes a different type, which is called microcytic hypochromic anemia, where the cells do progress through this step. So there are no nuclei, and the cells are small, but in this case, hemoglobin is not being incorporated properly into the cell structure, so they end up being smaller and not as red as typical erythrocytes. To treat and prevent neural tube defects, it's recommended that pregnant women supplement with 400 micrograms of folic acid. So that's double really the recommended need for the age range, but this is thought to accommodate the extra DNA synthesis needs during pregnancy. And this is really important, especially in the first 3 weeks when neural tube defects are more likely to occur. Ever since the U.S. started promoting folic acid supplementation at this rate, we've actually seen a 70% decline in spina bifida, which is a really common form of neural tube defect. Folate is overall not a very toxic nutrient. But we do have an upper limit, and that's only for synthetic, so supplemental forms, and the fortified forms, so like in cereal grains and flours. The upper limit is 1,000 micrograms, and the reason we have this set is because if you exceed this amount, it makes it really hard to recognize if someone is becoming deficient in B12. And so folate can actually mask B12 for a long time, mask B12 deficiency for a long time, because the folate will be able to still form red blood cells because you have plenty of folate always coming in to produce those new DNA bases, but none of it's getting passed over to homocysteine through B12 if B12's not there. So it's getting trapped as 5-methyl, but there's always more folate coming in to make the other forms of folate for red blood cell and DNA formation. So really it's not because there are toxic symptoms, it's really just to allow us to detect a B12 deficiency early on. The reason that high folate intake can mask B12 deficiency is because the folate will alleviate that megaloblastic anemia. And B12 has other effects, particularly on the neurological system, and so those can take months to years to develop. And so the damage to B12 deficiency on neurological systems would then go undetected, and actually can be hard to repair if it's allowed to go on for too long. So the tolerable upper intake limit of 1,000 micrograms then just allows us to be able to detect those neurological symptoms earlier and actually intervene and reverse them. Okay, so that brings us to the end of folate slides. So I think we'll stop here for today, and then we'll pick up with B12 for Monday. And since these two nutrients are so intertwined, we'll kind of review a lot of the same things again with folate. And if there are any questions, then please be sure to ask those in the B12 lecture.